Hello, everyone. This is Jianming from GemPharmatech. Welcome to today's webinar. So in today's webinar, I'm going to discuss with you about T-cell engagers, and starting from in vitro design of molecule to in vivo efficacy and safety evaluation. We know that in the past 4 to 5 decades, cancer immunotherapy has received increasing attention with the success of several molecules. So the monoclonal antibody-based therapeutics holds great promise to fight against cancer. However, such therapeutics, monoclonal antibody-based therapeutics, also face several challenges. So first of all, in order to have an effective function against cancer, the effector immune cells have to be infiltrated well into the tumor, which is not trivial, especially for solid tumor. So due to a very tight connections in the extracellular matrix and also the tumor mass, for many solid tumors, the infiltration of immune cells is very poor, and this could lead to a poor response to therapeutic drugs. And even after the immune cells have successfully infiltrated into the tumor, the cancer cells are in general immunosuppressive. That's why the cancer cells can grow in vivo. So the infiltrated immune cells has to deal with immunosuppressive environment. And even after successful binding with the therapeutic antibodies, we know from history that monotherapy are not very effective for cancer cells. So they develop various mechanisms to become resistant to cancer therapies. So as we can see on the right-hand side, so currently most of the monoclonal antibodies which have successfully entered the market, they actually focused on a couple of molecules, mainly centered at the PD-1, PD-L1 axis, and also the CTLA-4 molecules. And many other molecules are still struggling, either in preclinical, and most of them are struggling in clinical stage. In the past 20 years, with the advance of technology, the emergence of bispecific antibodies opened a new chapter for cancer therapy. So those molecules are very powerful, not just because they combine two monotherapies together, but because they have different structures and different combinations. So many of them can give you a synergistic effect depending on how you select the different molecules. Overall, those bispecific molecules, they target two different antigens. These two antigens can be present on the same cells, or many cases, they are present on different cells. So one from the cancer cells and one from the immune cells. So those bispecific antibodies are able to redirect the immune cells targeting against cancer. However, due to the complex nature and the structure of those antibodies, many of them actually demonstrated immune-related adverse effects, especially in clinical trials. So here I listed so far approved bispecific antibodies. And as we can see, many of them are still targeting hematological cancer. Although there are some new players which are very effective against solid tumor, but still most of the currently approved bispecific antibodies, they are more effective towards hematological cancer. And those are the commonly found adverse effects in clinical. Many of them are actually immune-related, suggesting that when designing those molecules, so based on the mode of action, it's very important also to have a powerful system, a preclinical system to evaluate potential toxicity. When I went through the published database, so I found there are more than 15,000 molecules currently either in preclinical stage or in different stage in clinical trials. This means this is a highly competitive area. And when I listed all the targets, all the molecules by targets, as you can see, there's one target which is champion of the champion. So this is the CD3 molecule. And those bispecific antibodies which targeting CD3 on one arm and another arm targeting tumor-associated antigen, we also call those bispecific antibodies T-cell engagers. And because of the vast majority of the bispecific antibodies are actually T-cell targeting antibodies. So for today's webinar, I will focusing on the T-cell engagers. So some people may think the bispecific antibodies or T-cell engagers are very easy because you simply combine two different monoclonal antibodies. That's actually not true. So in the next few slides, I'm going to share with you a story published story to guide you through that proper designing of the molecules is very important in order to achieve success in preclinical evaluation. So in this story, the authors, they screened a CD3 targeting monoclonal antibody with different affinities. And they combined these different affinity CD3 antibodies with tumor targeting antibodies to form a bispecific antibody. So first, they evaluated the internalization of the bispecific antibody. And to do this, they designed a molecule. So they combined two different fluorophore with a peptide. In this peptide, there's a cleavage site for the cathepsin B. So when this molecule is outside of the cells, so the energy from the Alexa fluoro 568 was transferred to another fluorophore. So this is the Alexa 647. So basically, you will get a color from this molecule. And once the entire molecule was internalized and entered the lysosome or late endosome, the peptide was cleaved by cathepsin B in low pH environment. Then these release the first fluorophore. So in this case, you will see a different color with a different fluorophore. And you can quantify the number of molecules which are cleaved, indicating the number of molecules which are internalized by the fluorescence intensity. So here we see the results. So the authors, they combined the different CD3 targeting arm with another tumor targeting arm. And you can see that they selected three different CD3 targeting antibodies. They have different affinity to the CD3 molecules. So this is incubation with human PBMC. And those molecules which has a moderate affinity to CD3 molecules are highlighted in black, and those which has a weak affinity to CD3 molecules are highlighted in dark gray, and those which has very weak affinity to the CD3 molecules are highlighted in gray. So you can see no matter which tumor targeting antibodies they use, so in all cases they observed the phenomenon that the antibodies which has the strongest affinity to the CD3 molecules are the most efficiently internalized by the PBMC. They also evaluated tumor killing effect in vitro. So here they use the cancer cells. So this is an ovarian cancer cell, OVCAR3. And they used for the TAA molecule, they choose the mucin 16. And they combined these tumor targeting antibodies with the CD3 targeting different affinity CD3 targeting antibodies. So as you can see, the molecule which has the strongest affinity to the CD3 molecule gives the most efficient tumor killing effect. And showing here in this curve, as well as the lowest EC50 value. And this actually correlated very well with the T cell activation. So here they simultaneously measured the T cell activation by the CD25, so using this biomarker for CD8 T cells. So this actually correlated very well. So the killing correlated very well with the T cell activation, and this both correlated very well with the affinity of the antibodies towards the CD3 molecule. So the CD3 binding affinity actually drives the T cell activation and the cytotoxicity in vitro. And for the tumor targeting antibodies, because they don't have different antibodies with different affinity, so they used another alternative approach. So they used the competing antibodies to block the TAA, which is the mucin 16, on the tumor cells. So as we can see here, with the increasing amount of the blocking antibody, which reduce the binding of the T cell engager to the tumor cells, we see that the killing effect was decreasing with increasing concentration of blocking antibodies. But for each concentration of the blocking antibodies, we can still see that the molecule which gives the strongest binding affinity to CD3 gives the best killing effect in all concentrations tested here. But does that mean that when designing a T cell engager, one should use the molecule which gives the strongest binding affinity to CD3 and to the tumor cells? So next, they performed a in vivo biodistribution assay. So they radioactively labeled the bispecific molecules, the T cell engagers. And here you can see the molecule which has the moderate affinity to CD3 molecules, although it shows superior cytotoxicity and the tumor killing effect in vitro. In vivo, this molecule is enriched not only in the tumor cells, but also in the spleen. So this means there might be potential toxicity when you give these antibodies to human patients. On the other hand, the molecule which has the weak affinity to the CD3 molecule, although the tumor killing effect was weaker in vitro compared to the molecule which has the moderate affinity to the CD3, but in vivo, it demonstrated very specific biodistribution. So most of the antibodies were detected only in the tumor, but very little in the other normal tissues. So this actually gives the advantage that with this molecule, with a lower or weaker affinity to the CD3 molecule, this would allow you to, for clinical administration of the molecule, this gives you the advantage that you can, when you are doing dosing escalation studies, you can give higher doses of these antibodies in comparison to the one which has a moderate affinity to CD3 molecule because of the biodistribution. So this molecule potentially would give you toxicity when injected in a higher concentration. So from this story, we learned that it is not so trivial to design a T-cell engagers. You need to take into consideration not only the efficacy, but also the potency and the potential in vivo safety. And the in vitro model actually can give you a very good estimation in these settings. At GemPharmatech, we established a one-stop solution for the evaluation of T-cell engagers. So starting from antibody discovery to in vitro evaluation of cytotoxicity and T-cell activation, so based on the mode of action of the molecule, and to in vivo evaluation of the anti-tumor efficacies, PK/PD, and the potential toxicity in preclinical stage. So in the next part, I'm going to talk about each part in more details. So let's start from the first part, which is the discovery and the in vitro characterization. So for this, we have generated a platform based on our humanized antibody humanized mice called NewMap. So in this platform, we are able to generate antibody molecules and in different formats. So we can generate conventional IgG molecules and also the heavy chain only antibodies, as well as common light chain antibodies. So the common light chain antibodies is also is frequently used by many other companies like pharma or biotech for the discovery of bispecific antibodies. So here I'm going to show you one case study where we generated a bispecific antibody, actually not only one, but several ones in different formats. So we compared our in-house generated antibodies to the benchmark molecule. So for these antibodies, we used the same TAA, so the same molecule which actually targeting the tumor cells, but we generated the target A antibodies on the T cells by ourselves. And here you can see we generated those antibodies in different format and different also with different valencies. And in comparison to the benchmarker molecule, you can see that our in-house generated antibodies showed comparable binding to the Jukat cells. However, in vitro killing assay, so when we tested the cytotoxicity of these antibodies with using a Jukat luciferase reporter assay system, here we can see that our in-house generated antibodies were more potent than the benchmarker molecule, as shown here. And also from here, you can see that the lower value of EC50 suggested actually higher potency of our in-house generated antibodies. And so once you have generated preclinical candidate molecules, either by yourself or you can outsource this service to us, the next part would be to evaluate the anti-tumor efficacies in vivo. For this, we have two commonly used mouse models to serve this purpose. So the first model we have is the humanized mouse model. Here we use either we use the immune cells either from peripheral blood mononucleocyte from healthy donors or using the cord blood derived CD34 positive hematopoietic stem cells to engraft severe immunodeficient mouse model. So here we use NCG. So NCG was developed by GemPharmatech, and we licensed NCG and its derivative strains actually to Charles River for the North American market. So the advantage of this platform is that because we use human PBMC or human HSC cells, so we are able to get human T cells. And since we use the immunodeficient mouse model, so this would allow the engraftment with human cancer cells, not only the human cancer cells, but also human cancer patient-derived material. So in this model, you would have the option or freedom to choose various human cancer cell lines based on the cancer indication you're interested in or the expression level of the target molecules on the cancer cells. However, the limitation of this model is that these humanized mouse model, although they are very powerful and we can get functional T cells, which is sufficient for the evaluation of T cell engagers. However, the entire immune system is not competent enough. So in comparison to the wild type mouse or to human beings, the immune system is not competent enough. Therefore, this model is more suitable for the evaluation of anti-tumor efficacies. But for toxicity evaluations, we recommend to use our CD3 knock-in mouse model. So this model is also known as the syngeneic mouse model. And it is the biggest value of this model is that it is based on an immunocompetent environment. So we knocked in the CD3 molecules into the wild type BABC mice. The mice will express human CD3 molecules on the surface of their T cells. And when combining these models with a murine tumor cells engineered to express human targets, so we can also use this model for both efficacy evaluation, but most importantly, we can also use these to evaluate potential toxicity, especially those related to the immune system. And for the humanized mouse model, so as I mentioned, we typically use two different strategies, so either PBMC or HSC. So the advantage of the PBMC engrafted model is that the engraftment took place very quick. So you can see 2 weeks after we injected the PBMC, we got more than 25% human CD45 positive cells in the peripheral blood. And when you do the PBMC injection in the NCG mice, most of the immune cells you are going to get are actually CD3 positive T cells. So this model is ideal for the evaluation of test articles which are targeting human T cells. So here I'm going to show you another case study. So this is a case where we helped our sponsors to select and evaluate T cell engagers before clinical stage. And the study was actually performed at GemPharmatech. So the target from our sponsor, so they developed T cell engagers targeting the CEACAM5, so the embryonic carcinogen antigen. And this is the format of the bispecific antibody or the T cell engagers. So they generated different molecules with different CD3 molecules with different affinity to the CD3 molecule. So as shown here, they have the parental CD3 targeting molecules and the two mutant version or engineered version, which showed different affinity towards the T cells. As you can see, the version 2 is the one which showed weaker binding to the CD3 molecule. As shown here, you can see that the activation of the T cells as analyzed by the cytokine release was much weaker and in a slow kinetics in comparison to the parental CD3 targeting molecules. So as we learned already from the previous lesson, that when designing a T cell engagers, it's very important to take into consideration the different aspects of the molecule. Therefore, the sponsor decided to move forward with further optimization with the molecule, focusing on the version 2. And in the next step, we also tested the tumor binding using different human cancer cell lines in vitro. So we tested the target expression using a flow-based assay, and also we tested the in vitro killing effect. And here we included both the positive control and the negative control. So the positive control was the T cell engager from Roche, and this is a negative control with the same CD3 molecule. But on the other arm, here we used a control antibody, which is not targeting the CEACAM5 molecule. So here we can see that depending on the expression of the targets on the cancer cells, we can see that those cells which has a higher expression of the target showed very good in vitro killing with both the CD3 molecule and also the molecule, the benchmark molecule from Roche. And although here you can see that the molecule from our client was actually more potent than the benchmark molecule. Then we also tested the in vivo, the efficacy in vivo. So here we used a cancer model. So here we used the LS174. So the colorectal cancer model, here we co-injected the PBMC with the cancer cells, then started antibody treatment roughly 1 week after the tumor cells and the PBMC implantation, and monitor the tumor growth. So this is how in the control group, how the tumor growth looks like. And here in the different, we tested all 3 different versions of the molecules. So the molecules with different CD3 molecules with different affinity to the CD3. And we can see that at higher concentration, so 3 mpk, all 3 molecules showed more or less comparable tumor killing effect. But at a lower concentration, so when the antibody concentration is limiting, so the one which showed weaker binding to CD3 was less potent in comparison to the other 2, which has a more stronger binding to the CD3. Then we also did a dose escalation studies using the version 2 molecule. Here you can see we obtained dose-dependent tumor killing effect. And last but not least, we combined the T cell engagers with a anti-PD-L1 antibody. So the Tecentriq, we use Tecentriq here. And as you can see, we were able to obtain a synergistic effect. So both Tecentriq and the T cell engagers as a monotherapy can inhibit the tumor growth, but the combination of the 2 drugs gives you much stronger tumor killing effect. And this actually correlated very well with the in vivo pharmacodynamics. So here we actually saw that in the combination group, more T cells, especially the CD8 T cells, which is the the most important one in the context of tumor killing, are actually highly infiltrated into the tumor. So we did the TUS analysis at the study endpoint. So this is a very nice story to demonstrate that how we use our models, we can help our clients, to from in vitro selection of preclinical candidate molecules and to the in vivo anti-tumor efficacy evaluation. Actually, using this PBMC NCG model, we are not only able to evaluate T-cell engagers for oncology indications, but we also have mouse models for autoimmune disease, as many of the T-cell engagers now are also very popular, not only targeting cancer, but also autoimmune disease. So here we have established a SLE model using PBMC donor from SLE patients. And here we tested different test articles, including the T-cell engagers, both bispecific and trispecific, and also CAR-NK molecules. The hallmark characteristic of the SLE disease is the secretion of the auto antibodies, such as the anti-double-strand DNA antibodies, which eventually leads to the disease progress. And here we use this as a biomarker. We can see that after giving the T-cell engagers, so both the bispecific, so the Blincyto and the trispecific ones, which are very effective in depleting or reducing the anti-double-stranded DNA molecules. And here we also did pathology analysis for those in those models. One of the biomarker in the clinical for the SLE disease is the deposition of immune complex in the kidney, which induce a nephritis. Here we can see in the G2 group, which is the vehicle-treated modeling group, we can see the deposition of the immune complex as shown by immunofluorescence. And after treating with different molecules, the immune complex was significantly reduced or cleared by the antibody treatment. However, there's a limitation concerning the PBMC NCG model, and the biggest limitation is very often you will get a donor-dependent GVHD effect, and this potentially would shorten the therapeutic window for your drug. So this is how a typical GVHD looks like. So the mice showed Hongqin and a pyeloerection. And when we did pathological analysis of different tissue organs from the mice, we observed significant immune cells infiltration into the different organs. And depending on the donor you are using, so in the worst case, a severe GVHD can reduce the survival or shorten the lifespan of the animal, and this actually makes the in vivo efficacy evaluation very difficult, because mice are dying not because of the treatment, but rather because of the GVHD effect. So to solve this problem, we used two different strategies. So the first strategy is we developed a mouse model called NCG MHC double knockout. So in this mouse model, we knocked out the MHC class 1 and class 2 molecules from the parental NCG mice. So as you can see here, we did a head-to-head comparison using three different PBMC donors, either in NCG mice or in NCG MHC double knockout mice. So when we injected the different donors into the NCG mice, you can see depending on the donor, we can observe a donor-dependent GVHD effect, as shown by the clinical observation here, like the GVHD score, the body weight, the survival. You can see that different donors may have an impact on the animal. However, when we injected the same donors into the MHC double knockout NCG mice, you can see that the GVHD effect was significantly reduced. And here we don't have any issue with survival, so that all the mice are viable up to 70 days after PBMC injection. And this was actually not due to the differential level of PBMC engraftment, because when we did flow analysis to look at the human cells engraftment, this is very much comparable between the MHC double knockout model with the NCG, the parental NCG model. So in a short word, the MHC double knockout mice can help to reduce the GVHD without compromising the human T cells engraftment. So we actually used this model system for to test it many T cell engagers. So here I'm going to show you one example. Because, as I mentioned, different donors can give you different effect. Therefore, in order to precisely evaluate the anti-tumor efficacies of T-cell engagers, at GemPharmatech, we perform in-house screening, pre-screening of the different PBMC donors we have, so that whenever there's a sponsor who would like to test their T-cell engagers molecules using our PBMC model, we can always recommend the most appropriate model. So here I use the tarlatamab. So this is a DLL3 targeting T-cell engagers as an example. We choose this molecule because of the very successful approval of this molecule by the FDA for the treatment of late stage and malignant metastatic small cell lung cancer. So we tested a couple of lung cancer cell lines, human lung cancer cell lines we have in-house. So we quantified the expression of the tumor target. Here we can show that there are 3 cell lines which showed a high level of DLL3 expression. And we decided to continue with these, with one of them. This is the SHP77. So here we used our MHC double knockout mouse model, and we injected the PBMC and the SHP, the tumor cells, not together separately, but on the same day. Then 2 weeks later, when the PBMC or the T cells engraftment was successful, we started giving treatment to the tumor model. So as you can see, in vitro, we can show that the benchmarker molecule, so the tarlatamab, actually can bind to the DLL3 positive tumor cells very well. So here we compared the DLL3 positive versus the DLL3 negative small cell lung cancer cells, and we observed very good binding. And for the in vitro killing assay, we incubated the tumor cells, so the SHP77, with our Jucat luciferase reporter cells. Here you can see that in the presence of the therapeutic T-cell engagers, the tumor cells, so the activation of the Jucat luciferase cells were very obvious. And we also evaluated the T-cell activation in vitro. So we selected different biomarkers for T cell activations. You can see that at different kinetics, different time points, those markers were activated by the T cell engagers. However, some markers are activated in a fast kinetics, but some markers are activated in a relatively slow kinetics. So that depends on the markers we are choosing. So for example, the PD-1 is an exhausting marker, which is well known to be activated at a later stage. So here we can see that the PD-1 was only activated significantly after 48 hours. And moving forward to the in vivo test, so we tested the anti-tumor efficacies of the tarlatamab using 4 different PBMC donors. So as you can see, different PBMC donor will engraft the mouse to a different extent. So some showed relatively higher level of T cells in the peripheral blood, but some had a lower T cell engraftment. However, in all cases we tested, so in all the 4 PBMC donor we tested, the tarlatamab were able to demonstrate very good tumor killing effect and in a very efficient way. So you can see that almost after 1 week, nearly all the we got complete response almost in every animal. However, when we closely look at the clinical observation, we saw that different PBMC donor, they had a different effect. So for some of the PBMC, for some donor, the body weight was relatively constant and the GVHD score was low. But there are donors which showed higher GVHD effect upon the T cell engager treatment, suggesting that these PBMC donor are actually sensitive to the T cell engager treatment. So it is actually very important to evaluate all the different aspects of the T cell engager molecules. And here at GemPharmatech, we offer different PBMC donors. So this would allow you to monitor potential anti-tumor efficacies as well as immune-related adverse effects using different PBMC donors. Another strategy to overcome the GVHD effect is actually to use our cord blood-derived CD34 positive hematopoietic stem cells engrafted model. So as you can see here, the major difference between the HSC versus the PBMC engrafted model is that in the HSC model, you do not only get T cells, but you also get B cells. And this could be actually helpful if your T cell engager molecules are actually targeting the B cells. So we can evaluate B cell depletion in this model without the extra inoculation of CD19 or CD20 or BCMA positive cancer cells. And another difference in comparison to the PBMC model is that the engraftment in the HSC model took much longer time. So as you can see, so depending on which model you are using and also the donor, so averagely speaking, for the HSC model, it would take around 8 to 12 weeks until the HSC is well engrafted in the animal. However, we understand that time is really limiting and critical for most of our clients. Therefore, at GemPharmatech, for all the HSC engrafted model, we produced those animal model in a rolling stock. This means we always do the engraftment in advance. So whenever you would like to use those HSC engrafted model, we have reconstituted mouse models ready to go. So you don't have to wait for 2 months until you can get enough animals for your project. And as you can see here, the HSC engrafted model hardly have any problem with GVHD. You see here the body weight, so and the survival curve. We monitored those animals, more than 120 days, so more than 3 months, and there's absolutely no issue with this model. So this is actually particularly helpful if you are dealing with a slow-growing tumor. There are some tumors which grow extremely slow, and using the HSC model gives you the advantage. Otherwise, in the PBMC model, you may see GVHD before you see anti-tumor efficacies with your test article. For the HSC engrafted model, we also had proven experiences serving different clients. So here I'm going to show you another case study. This is also a collaboration with one of our clients, and the study was performed at GemPharmatech and published also in a peer-reviewed journal. So here we evaluated a tri-specific T-cell engagers. So here, in addition to the commonly used anti-CD3 molecule, the authors also they added another anti-CD28 as a co-stimulation molecule to the T-cell engagers, potentially hoping that this could give you advantage in boosting the T-cells against tumor. And you know that since here we have not only the CD3, but also the CD28. So we can't use a syngeneic model. So our CD3 knock-in mouse models are not suitable for the evaluation of those molecules. Therefore, we choose to use our HSC engrafted mouse model. And also because the client, they are interested in observing not only anti-tumor efficacies, but they also want to look at potential toxicities. Therefore, we use two different models. So the first is the subcutaneous model. Here we used the Raji luciferase lymphoma cell line injected into the HSC humanized mouse model subcutaneously. And here we can observe a dose-dependent tumor killing effect. You see, for the tri-specific antibodies at the highest dose, this almost resulted in a tumor-free after 3 weeks of treatment. Then in the second model, we also tested a systemic model. Here we used NAM6 luciferase cells IV injected into our HSC humanized mouse model, and we observed the tumor growth via bioluminescence imaging, which is a non-invasive technique to monitor the tumor growth without the need to sacrifice the animal. And also we observed the survival of the animal. So as you can see, without any treatment in the vehicle group, actually the tumor cell inoculation actually leads to death of the animal. However, when we give the treatment of the tri-specific antibodies, we not only observe a reduction of the tumor growth, but also a significant prolongation of the lifespan of this model, suggesting that this molecule is very effective, and the animal actually benefit from the treatment demonstrated both by the tumor growth as well as by the overall survival of the animal. Here is the bioluminescence picture you can see. So to give a short summarize on this topic, so we have developed NCG, so the severe immunodeficient mouse models for the evaluation of T-cell engagers. However, we also noticed that currently there are different kind of molecules which are in preclinical stage under research and development, and many molecules, as I mentioned, like the tri-specific antibodies I showed you, which is not only targeting the CD3, but also other molecules or receptors, maybe not only on T cells, but on other immune cell types. Therefore, there is a demand to have different humanized mouse model, which supports the engraftment and development of different population of immune cells. So for example, in a recent published Cell paper, they developed this multi-tag. So these are also engagers, but they are targeting not only T cells, but different type of immune cells. So here they used this EGFR targeting tag, which not only targeting the EGFR on the tumor cells, but CD3 on the T cells and the PD-L1 on the dendritic cells. So there they used our humanized NCG, our 15 mice, demonstrated as a proof of concept study to demonstrate the successful anti-tumor efficacies of these multi-targeting molecules. So at GemPharmatech, we offer all these kind of models. So depending on which immune cells you are interested in, we can recommend different NCG or NCG derivative strains, which can support which can support the development of different immune cell populations. So in the last part, I will quickly show you the mouse models we have for the safety evaluation. So here I want to introduce to you our syngeneic mouse models. As you can see here, we have developed a lot of knock-in mouse models. Not only we can generate those mouse models not only with single target knock-in, but also double, triple, and even quadruple target knock-in. So those multi-target knock-in mouse models are particularly helpful for combination therapies. And one thing I would like to highlight is that for many of those mouse models, we generated them in a dual genetic background. This means we have many of those mouse models, both in the BLAC6 as well as in the BARBC background. So the reason why we need to use a dual genetic background is because we know that these two different backgrounds, they give a skewed immune response. So these two different genetic backgrounds, they not only differ in the MHC molecules, but also in terms of immune response. Those genetic backgrounds actually they showed a skewed immune response. With the BLAC6 mice, they are more towards a Th1 type of immune response with high level of IL-12 production, while the BARBC mice are more specialized towards the Th2 type of immune response. Therefore, we know that many of our autoimmune disease models, you can only create those models using the BARBC background. And one thing I also would like to highlight is that if you still remember, in the very beginning, I told you we have this humanized antibody mouse. That mouse was also generated based on the BARBC background, because these mice are very good at evaluating antibody-based reactions. So they are more competent to generate a more complex repertoire of antibody molecules, but also for the evaluation of the T cell engagers, which has different structures which might which may have higher immunogenicity and create immune-related adverse effect. So this is how the T cell receptor looks like, and we not only knocked in the epsilon subunit of the CD3, but all three subunits, so the epsilon, delta, and gamma, because there are different molecules, they're actually targeting different epitopes of the CD3 molecules. Therefore, in one go, we generated a knock-in mouse model where all three subunits from the CD3 molecules were replaced with human counterparts. So in this mouse model, the highlight of this model is that it expresses, instead of murine CD3 EDG, but rather human CD3 EDG molecules. And the endogenous mouse CD3 molecules have been knocked out. And we have validated the human CD3 molecule expression in different immune cells, in T cells in this mouse. And the immune cell profiling showed that the knock-in of human CD3 molecule does not change the immune cell population of these mice. This means the immune system was not perturbed by the knock-in procedure per se. And the in vitro assays showed that antibodies, so anti-human CD3 antibodies, were able to activate T cells from the CD3 knock-in mouse, but not from the wild-type BABC mice. Both for so the CD3, anti-human CD3 molecules were able to activate T cell proliferation as well as T cell activation showed here by the different biomarkers expressed on the surface of the T cells. And this is specific to the human CD3 knock-in mice, but not to the wild-type mouse. In vivo efficacy studies using a human CD20 engineered murine tumor cells combined with our CD3 knock-in mice, we were able to show that benchmarker molecules, so the mosunetuzumab, which targeting the human CD20 and the CD3, showed very good anti-tumor efficacies with minimal perturbation on the body weight and other clinical observation. So last but not least, we used this model for the evaluation of anti-tumor efficacies as well as potential toxicity. So this is another story. We actually collaborated with our sponsor, and the work was actually published also on a peer-reviewed journal. So here, our sponsor designed a T-cell engager molecule which targets the anti-PDL1, the PDL1 molecule on the tumor cells and the CD3 molecule. So they also designed another two monoclonal antibodies as a monotherapy. Here you can see in comparison to the UCHT1, so the molecule which targets only CD3 alone, the bispecific antibody or the T-cell engagers showed a much significant anti-tumor effect. And more importantly, when we evaluated potential cytokine release syndrome or potential immune-related toxicities, we compared these T-cell engagers, so the PDL1, CD3 T-cell engagers, with a benchmarker molecule. This is also from Roche. We can see that our client's molecule showed a much less pronounced cytokine release when treated with the T-cell engagers in comparison to the benchmark molecule. And this is also evidenced by the clinical observation, the body weight change of the animals. You see that when animals were treated with the benchmarker molecule, we observed significant or quite severe body weight loss of the animal due to the T-cell engager treatment. However, in the client's molecule, the body weight decrease was way much less pronounced, suggesting that this molecule is much safer. So when you do dose escalation studies in human patients, so this would allow you to give a much higher dose in comparison to the benchmark molecules. So here are some of our resources available to be coupled with our syngeneic, so the CD3 knock-in syngeneic mouse models. We have engineered quite a couple of murine cancer cell lines to express the human target. And also we intercrossed our CD3 knock-in mice with different either immune checkpoint or tumor-associated antigen knock-in mouse models. So in those double or triple knock-in mouse models, so this would allow you to not only to evaluate potential toxicities coming from the CD3 targeting arm, but also you can evaluate on-target off-tumor toxicities if the tumor-associated antigens are also expressed in normal tissues. So you can use those mouse models for the evaluation of both toxicities. So that's all for today's webinar. Thank you for your time and for listening.